All right, we're starting now then. Good morning, everyone. I welcome you all for today's Center Seminar Series. Today we have with us Dr. David Thal from MIPS. I would like to begin by acknowledging the Wurundjeri people of the Kulin Nations as the traditional owners of the land on which our Parkville campus stands. I also acknowledge the traditional custodians of the various lands on which you are all joining me from today, and I pay my respects to their elders, past, present, and emerging, and the Aboriginal elders of other communities. I'm Meenakshi, a Center PhD student and the Monash University. Today we have with us Dr. David Thal from MIPS. Dr. Thal is an NHMRC Investigator Fellow and a co-leader of the Analytical and Structural Neuropharmacology Group in the Drug Discovery Biology team in MIPS. Dr. Thal received a PhD in chemical biology from the University of Michigan, where he received a training in structural biology from the lab of John Chasma. And then in 2011, he moved to Australia to begin his postdoctoral research under Arthur Christopoulos and Patrick Saxton at MIPS, which also included a year-long training in the Brian Kobilka lab in Stanford. After completing an ARC-DCRA fellowship in 2019, he established a research group within MIPS that focuses on determining the molecular structure and function of proteins, with a particular emphasis on understanding novel paradigms of drug actions, drug design that are therapeutically relevant. Welcome, David. And before starting, I would just like to request everyone that is joining over Zoom, please stay muted throughout the seminar, and we'll be having a Q&A session later on. And if you have any questions, you can just type in the chat box. Welcome, David, and over to you.
Thank you for that introduction, and hopefully everyone can, sorry, just arranging my Zoom windows here. Hopefully everyone can see the slides. So thank you also to the CCE-MMP for the invitation to present some of our research today. This is going to be a talk that's a little bit unusual from the normal talk that I'm going that I would normally present, and that's I want to give a series of short stories about our adventures in structure-based drug discovery, and particularly sort of the ups and downs. So there'll be sort of a few projects that are sort of on sort of the ups, if you will, a few projects that are sort of in the bottom, waiting to take off, and sort of a few that are kind of stuck in the middle. So I just want to start with sort of the definition of structure-based drug discovery, at least according to the internet, and that's the design and optimization of the chemical structure of a drug based on the structure of its biological target. Now, if you came to this, sorry, it's a little bit laggy there. So if you were coming to this seminar hoping to get a really nice, elegant story about structure-based drug discovery based on this definition, then I'm sorry to disappoint you. Rather more, what tends to happen is a scientist somewhere, this could be a medicinal chemist or a biologist, goes, "Hey, we've made this exciting discovery. Can you, as a structural biologist, help identify where this small molecule may bind into our favorite protein?" Or it might be that they have a newly discovered protein that's important for a particular therapeutic target, and at least prior to AlphaFold, there'd be no good structures. And so generally, as a structural biologist, we're trying to help people in sort of this early stage drug discovery process. Now, I don't think I'm probably preaching to the choir in the sense that the impact that structural biology has had on drug discovery, and particularly with the approval or its impact on the FDA-approved drugs, as recent reviews estimate that nearly 90% of FDA-approved drugs have been impacted by structural biology research that has been deposited into the PDB, and there's been some relatively nice reviews that sort of highlight that process. So today's really going to be just a collection of a few short stories about our interest in using Cryo-EM to facilitate structure-based drug discovery. First, I was going to start with a few words about myself. Then sort of the first short story will be our sort of a foray into Cryo-EM with the enzyme 12-lipoxygenase. Then I'll talk about a new project that we've been working on, which is sort of the quest for better male contraception. And then finally, sort of the bread and butter of our group, and that's where the allosteric modulators are. So I did my PhD at the University of Michigan under John Tessmer, being trained as a crystallographer. And really, I just wanted to present this as examples. This is where my interest in structure-based drug discovery was probably initiated. And that was, we were interested in trying to design small molecule inhibitors of a particular kinase, in this case, the G protein coupled receptor kinase 2. During the course of the PhD, I was eventually able to solve a structure with this inhibitor bound into the active site of the enzyme using X-ray crystallography. And I'm really showing this for two reasons. One, I can't remember the last time I've actually seen a figure published that uses stereo view. And for those of you that don't know what stereo view is, you might be able to stare at this figure and cross your eyes and maybe see this in 3 dimensions. I'm not sure how well that will work on a monitor. But I also wanted to highlight that sort of the pros and cons of X-ray crystallography, and for better or worse, that's sort of the relationship between the experimental model and the X-ray or electron density map. And this gives you the advantage of that you can sort of remove part of the experimental model, recalculate electron density map, and you might get back a map around the region from which you removed. And this is a really powerful form of experimental validation in X-ray crystallography, though it can be biased if you're not careful. And I really just want to illustrate that as coming from an X-ray crystallography background, it's really nice having sort of this tool available. Also wanted to use this example as if you really are interested in structure-based drug discovery. John has been carrying out this work further, and there's a review that actually just came out last week that very elegantly shows all the work they've been doing on sort of kinase inhibitors using structures to facilitate the design of more potent and more selective inhibitors. So if you have an interest in that area, it's definitely a review that is really, really nice. In 2011, I came over to Australia to work under Patrick Sexton and Arthur Christopoulos. And really the idea wasn't to do any structural biology at all. I actually had enough of it and just wanted to do molecular pharmacology. Fortunately, the right set of circumstances came about, and Patrick and Arthur wanted to get into structural biology. I was quite fortunate to get involved in some early X-ray crystallography experiments and sort of establish X-ray crystallography of GPCRs here at MIPS, and this was relatively successful. Around the same time, Cryo-EM was starting to become mainstream or emerge more. And really, to Patrick, Denise, and Robert's credit, they helped bring Cryo-EM to GPCRs, and that's been relatively successful. And around this time, sort of realized that Cryo-EM is the way, and so we wanted to get onto it. In particular, we were interested if we could use Cryo-EM to facilitate structure-based drug discovery, and that's really where the stories I'll talk about today come from. Now, the first project was really our sort of our entry point into Cryo-EM, and that's with the enzyme 12-lipoxygenase. And this was really started by Elisa Glukova, who was at MIPS at the time, and we were sort of co-running a lab together at the time. She met Mike Collins from the University of Michigan at a Gordon Research Conference on GPCRs. And this project kind of begins as all projects do. Mike went, hey, I have this small molecule inhibitor. It's called ML355. It inhibits this enzyme 12-lipoxygenase. By the way, there's no full-length human structures of this enzyme. It's an important drug target for heparin-induced thrombocytopenia, where there's no good therapeutic options for treating that. And this drug has recently gone into phase 1 clinical trials. Do you think you could help us get a structure of this ligand bound to the enzyme? And of course, we like it a good challenge. We of course said yes. I'll also add that recently, maybe a couple of months ago, this drug ML355 passed the phase 1 trial and is now being fast tracked by the FDA. into phase 2 trials for the treatment of heparin-induced thrombocytopenia. I just wanted to give some acknowledgements to the team. Most of this work was really, Mike and Ted. So Mike was the sort of pharmacologist who did all the pharmacology related to this project. Ted Holman from UCSC is sort of the expert in lipoxygenase biochemistry, enzymology, and it had been trying to get structures of this 12-lipoxygenase with this inhibitor for quite some time, but wasn't having any luck. So that's where we brought in our team. That included myself, Elisa, Jesse, and Katrina, and most of this work was done out at the Ramachadi Center with the help of Hari. Now, just a brief word on lipoxygenases. I don't want to get into too much of the biology, as really today is more focused on the ligands that bind these enzymes. But lipoxygenases are a family of enzymes that oxygenase fatty acids, and this can be at various positions depending on the lipoxygenase. So 5-LOX will oxygenate the 5-carbon, 12-LOX, 12-carbon, and 15-LOX, the 15-carbon. The products of these are signaling molecules and are involved in various signaling pathways. For those that are wondering, there is a link to GPCRs and why this project was brought up at a Gordon Research Conference that was on GPCRs, and that's that at least in platelets, the activation of various GPCRs can lead to intracellular calcium, which activates phospholipase A2, resulting in arachidonic acid, which is the substrate for 12-lipoxygenase. This, of course, makes the metabolite 12 heat, and this can activate platelets. Interestingly, this product can also go on and activate other types of GPCRs. And so for those that might have been worried, there's a very clear link to GPCR signaling. But really the focus of this story was their discovery of a relatively potent and selective inhibitor for the enzyme 12-LOX, and we were really trying to help them get a structure of this and the enzyme in general. The purification scheme for the 12-lipoxygenase was relatively straightforward. We expressed it in HEK 293S cells. The protein had a his tag on the end of it. We did a nickel affinity purification and then purified it by size exclusion chromatography and got this, what you can see is a rather interesting looking set of peaks. And that's because this enzyme, depending on how it's purified by the phase of the moon, it can oligomerize into different forms. But almost always we at least saw some amount of tetramer and some amount of dimer. And I'll say that in the purification from the results that follow, this was done with ML355 added throughout expression and purification at the concentrations that we could use. Now, as I mentioned, coming from an X-ray crystallography background, our first interest was actually kind of get crystal structures of this. We had to go at that for maybe a week or two and went, you know what, this protein actually might be big enough for Cryo-EM. So Lisa went and did some negative stain, and the protein looked suitable. And before we knew it, we were able to get some relatively high resolution structures of 12-lipoxygenase. And so this is just showing sort of the workflow that we did. This is, I guess, a pretty standard workflow. We collected around 7,000 movies on a Titan Krios, multiple rounds of 2 and 3D classification, a bit of refinement in CryoSpark, and we were eventually able to get a 1.8 angstrom structure of a tetramer, which we could then do symmetry expansion and bring it down to around 1.7 angstroms. So we're pretty excited about that. This is a tetramer, so basically it's a dimer of dimers. I'm not going to get too much into sort of the secondary and tertiary structure of the enzyme, other than it was really high resolution, so we were able to accurately model all the various amino acids, which was really nice. However, we looked at the active site of the enzyme, sort of highlighted by these sort of red circles. We saw a blob in the active site that was far less convincing, particularly for a high resolution structure. The density just really didn't make any sense. It was symmetrical, it was discontinuous. It looked like there could be 2 head groups. We really weren't sure what was going on. We tried modeling ML355 in there a few times, and you could maybe convince yourself it was bound, but realistically we were fooling ourselves. So we weren't really quite sure what was going on there. We did a bit of 3D variability analysis, looking at the sort of variability of the collected data. And what you can see is there sort of a bit of dynamics between sort of the various dimer of dimers, sort of shown here. Now, the overall dynamics sort of aren't really important, but what was important was looking at the active site. And if we zoom in on the active site, what you can see is that in the first class, uh-oh, I ran out of, guess we're not going to play today. In the first class, what you can see is that the ligand was bound probably to one of the monomers. And as the movie progresses, you can kind of see the density shift over to the other monomer. And we thought that this could be potentially a break in the symmetry, and that maybe it was 50% occupied to one side and 50% occupied to the other. Whereas if we looked at the density sort of in the other classes, it was relatively stable throughout, and it didn't seem like it was really moving between. So what Jesse went back and did is he went into did some more 3D variability analysis and cluster and sort of the cluster mode, and he was able to pull out a subset of molecules that had clear ligand density in the active site. And that density sort of looked like this, relatively high resolution. Well, we could start to make out that there was a lipid tail, and there were some sort of blobs that we weren't quite sure, other than it clearly wasn't ML355. We were sort of having a guess that this looked like phosphates, maybe some sort of sugar. With the help of lipid maps and sort of investigating the various types of lipids, we were able to work out that it was basically a fatty acid, acetyl-CoA, that was bound in the active site. As you can see, fits the density very, very clearly, and a very nice fit. If we look at sort of how the dimer was situated, what we would see is that we'd have one molecule, one monomer would be fully bound to the molecule, and then the other monomer, there's just sort of a little bit of discontinuous density that we weren't quite sure what that was from. So we went and tested this in some activity assays. So Ted tested allesyl-CoA and actually found that it is indeed an inhibitor of 12-lipoxygenase. This is at relatively high concentrations with an IC50 around 30 micromolar, but the concentration of fatty acid CoAs can increase up to 100 micromolar under certain metabolic conditions. We're still trying to work out the exact length of this chain and whether it's allesyl or palmitoyl-CoA, and that's some work we're trying to currently do either by mass spec or through inhibition assays. But this wasn't the first time that someone had this observation. We found a paper back from 1995 where a group had shown that fatty acid acyl-CoA enzyme esters can inhibit lipoxygenase and rabbit platelets. What this actually means in terms of biology, we're not quite sure. And that's really sort of trying to work out what's going on here in the future, other than I think going into this project, none of us had any expectation that we'd end up with a acetyl-CoA fatty acid bound in the active site of the enzyme. Now, the story doesn't really end there. I'm mostly focused on what we observed from the tetramer peak. If we went to the dimer peak and did Cryo-EM on that, what Elisa actually found was you could get 4 structures from the same 12-lipoxygenase dimer grid. You can get a monomer, you can get a dimer, you can get a tetramer, and you can get a hexamer. And now things became even more confusing. We suspect that the dimer is probably the more physiologically relevant conformation of the protein. And this is certainly the one where we can see sort of an open and closed form of the active sites, which mimics what is more closely seen in other lipoxygenases. Whereas in the tetramer and hexamer, they're all sort of in this open conformation. There could be some sort of regulatory mechanism that's going on in that, in the dimer, the active sites are able to sort of interact with, or in a position where they could interact with the cell membrane, which is where arachidonic acid would be. Whereas in the case of the tetramer and hexamer, the active sites are relatively buried and not readily accessible. So there could be some sort of regulatory mechanism that's going on. Suffice to say, through a lot of work, we've got a lot of different structures of 12-lipoxygenase in various conformations, both that we've done with ML355 and without, and then we sort of see the same thing across all of these, and that to date, we still don't have any structures of 12-lox with ML355. So we're still trying to sort of wrap up some of the biochemistry and our understanding going on. What we got were some really cool structures with a really interesting lipid bound to them, but not really what we wanted at the start. The next project that I was going to talk about is sort of the quest for better male contraception. This is really a project that it has been led by Sabatino Ventura at MIPS. Almost as long as I've been at MIPS, Sab has approached me at Beer Club and gone, hey, when are you going to solve the structure of P2X receptor, P2X1? And the answer was always, well, yeah, maybe someday. Finally, with Cryo-EM, and we got a really good student, PhD student, Felix Bennett, so we decided to give this a go and this involves the other members of the team, the Cryo-EM team, Jesse, Lisa, and Hari. So hopefully I don't have to give too much of a background on unintended pregnancies and contraception. There's 120 million unintended pregnancies that occur globally each year. And this is due to a lot of different reasons I'm not going to get into. But one of the main ones is that for women, there's lots of different forms of contraception that are available, whereas for men, it's mainly condoms and vasectomy, with the former having a relatively higher rate of failure, and the later being less reversible. So there's less of a reason that people might want to get a vasectomy, particularly when they're in their earlier years. And so there's a definite need to try and get some form of male contraception that could mimic the likes of a pill. And that's really where the P2X1 receptor comes into play. It's a genetically validated co-target for male contraception. It's a trimeric ligand-gated ion channel. So as a monomer, it has two transmembrane spanning domains and an extracellular domain. Together, these form a trimeric ion channel that is activated by ATP and allows the permeation of cations. Now, in terms of male contraception, what we would like is a P2X receptor antagonist, but the discovery and development of P2X1 receptor ligands has been rather difficult. And so we're hoping to get a structure to try and facilitate some alternative approaches. So for the purification of the P2X1 receptor, we've essentially used the full-length wild-type human receptor, with the exception that on the C terminus, we put a 3C cleavage site, added green fluorescent protein and a hexahistine tag. We express this in insect cells, solubilize with detergent, purify by metal affinity chromatography, and eventually size exclusion chromatography. And I'll just note that at least in our initial work, we weren't adding ATP to the purification because we were trying to get a ligand-free conformation of the receptor. Initial purifications weren't terribly successful, but with a bit of changes to sort of our purification strategy, including changing the detergent to LMNG, adding glycerol, and doing 2 rounds of metal exchange chromatography in between a round of overnight 3C cleavage. We were able to get very nice pure monodispersed protein at relatively large quantities. You can confirm this by negative stain EM. That we did indeed have the P2X1 receptor. So then we started to optimize for cryo, and sort of our first attempts at that, what we realized is that the P2X1 receptor exhibited severe preferential orientation when Richard tried on grids, where all we really saw were use of the receptor from the top. So we tried a few various strategies to get more side views of the receptor. One of the first ones was using graphene oxide grids, which is an approach that requires minimal protein. And we were successful in getting more of the side views of the receptor, which is what we were interested in. But we were never really able to get this to higher resolution. And that's kind of a work in progress, I think. But what did work was adding a secondary detergent, which was fluorinated phosphocholine 8, and greatly increasing the concentration that we purified our sample at. There's a little bit of a balance in this, in that adding more secondary detergent increased the side views of the receptor, but it came at the cost of the particles preferring thicker ice. And so there's a bit of a balance between getting the right amount of secondary detergent into the sample. This is just sort of showing some of the initial sort of blobs that we got from our data collection. I see in that if all you have is views from the top of the receptor, you don't really get any sort of sensical structure. Once we started adding in a little bit of detergent, we started getting more of these side views and more evidence that we had a the full receptor. Eventually did a large-scale data collection on the Arctica out at Clayton. No ATP was added, and we were able to get to an angstrom structure, which we were actually quite pleased with. However, what we ended up finding was ATP was bound in the structure, and this might not be a surprise as it is activated by ATP, but there's also some evidence from one of the other P2X receptor subtypes that some of the subtypes, and P2X is one of them, can be quickly desensitized by ATP. So it's likely that it was quickly desensitized by ATP when it was in the cell and sort of locked into this desensitized state. So we thought we'd go back and try to get a higher resolution structure on a Titan, this time adding ATP to really make sure that it was present. And the structure went to relatively high resolution, which was great as it really facilitated modeling of residues within the transmembrane region of the receptor, which was a little more difficult at a slightly lower resolution. The structure of the P2X receptor looks similar to other P2X receptor subtypes. That is that it has sort of this trimeric transmembrane domain and this large extracellular domain where ATP binds quite nicely. Sort of has this chalice-like shape, if you will. And we observe 3 molecules of ATP bound in each of the orthosteric ligand binding sites, as well as what we believe is a magnesium ion coordinating all of the negative charge of the phosphates of ATP. But we can't see the intracellular regions of the NRC terminus, as these are disordered. So the next steps for this project are really characterizing the P2X1 receptor binding sites, so going back and looking at some of the residues around the ATP binding site and mutating them to confirm that they are indeed important for ATP binding. But what we really want to do is develop and validate some of the other P2X receptor antagonists. And so there's a collection of commercially available antagonists that we're currently pharmacologically characterizing, and some do look suitable for structural biology work, and hopefully we can then try and start working out where these bind using Cryo-EM. So stay tuned for more progress on this project. And if you really want to hear the whole story of this project, Jesse Mobbs will be presenting this at the ASCAT conference later in November. And so the last half of the talk today, I'm going to try and focus on a couple of projects that are really sort of the bread and butter of our laboratory, and that's looking at GPCRs and trying to understand where all the allosteric modulators are. So this is a theme of research that I co-lead with Celine Belant and Arthur Christopoulos in the Analytical and Structural Neuropharmacology Lab. There's a lot of postdocs and PhD students who have contributed work to the project that I'm going to present. A lot of people have done pharmacology and support these projects that I'm not even going to talk about. So there's a lot of people who've been involved in this project. It's also been a very collaborative project. Initially, there's a lot of collaboration with Patrick Sexton, Denise, Matt, and Rado from the University of Tokyo on collecting some of the data sets for some of the GPCR targets. Peter Skemmels and Moela and all the chemists associated with them have been instrumental in supporting these projects. We have really good collaborations with some experts in liquid simulations, including Ying Wang from the University of Kansas, Ron Jor from Stanford. Elise has also been helping out with some of the EM support. And then on the more translational side, we've been working with Chris Felder and Karuna Therapeutics and collaborations with Andrew Tobin at Glasgow. So I'm just going to give a very sort of basic overview of GPCR signaling, as I'm sure most people who have been in the CCE and MP seminars for a while have heard quite a lot about GPCRs. And that's really that they're activated by sort of an endogenous or orthosteric ligand. This can activate a receptor and cause receptor signaling. We can sort of characterize the binding of the ligand by its affinity and efficacy. I'm not really going to talk about that much further today, however. What's important to know is that GPCRs have allosteric sites, and these are sites that are distinct from the orthosteric site. And these are interesting as these are sites that are less conserved than the orthosteric ligand binding sites. So these represent a means to generate ligands that are selective. They're also interesting because they can modulate the activity of the endogenous ligand. So they can either increase the affinity of the endogenous ligand or decrease it depending on the type of modulator that it is. They can also alter the signaling properties of the endogenous ligand, and they can also activate and signal through the receptor on their own. And in essence, we like to describe them as operating kind of like a dimmer switch, in that they can kind of dial up or turn down the activity of the orthosteric ligand. Now, for the purpose of this talk, I'm mostly going to focus on, and I should also mention that allosteric modulation is sort of a major paradigm in drug discovery, not just for GPCRs, but drug discovery in general. But I'll be focusing on more, and let's talk about where these allosteric bonding sites are. This is just sort of a figure from a review we did in 2018, looking at where all the small molecule allosteric ligand binding sites are across the class A, B, and C GPCRs. And what you can see with the class A is that they bind to a number of sites sort of all around the receptor. What's interesting is I think almost all of these structures were determined using X-ray crystallography, as this is really before Cryo-EM was taking off. And I think most of these structures, not all, but most of these ligands or allosteric modulators are what you could call allosteric antagonists or negative allosteric modulators. And these are compounds that stabilize the inactive state of the receptor. And that's because from an X-ray crystallography perspective, determining structures of the inactive conformation was the easiest, if you will. And so that's why there's an overrepresentation of structures with allosteric antagonists. Since Cryo-EM has sort of become more mainstream in the field, there's now been an increasing number of allosteric modulator structures determined by Cryo-EM. And this is just highlighting some of the various recent examples. And in contrast to the crystal structures, what we actually see is more of these structures are with positive allosteric modulators, and that's because generally when we get structures with GPCRs using Cryo-EM is we're getting structures through the complex with the G protein. So we're looking at ligands that stabilize the active conformation of the receptor. Now, one particular example that came from the lab was with the A1 adenosine receptor. This is some work that was published last year, so I'm not going to talk too much about it other than to say we had to do quite a lot for the validation of where this allosteric modulator bound. This is MIPS 521. It bound outside of the transmembrane bundle into sort of this little pocket just above an activation motif around TM7 or 6. It's kind of a relatively small pocket. Here's a look at the EM map density, suggesting that there is in fact a ligand bound. We did a bunch of mutagenesis of the key residues around this binding site, showing that mutation of these residues can decrease the affinity of the allosteric ligand for the receptor. But perhaps more importantly, they would affect its allosteric properties. So there were a number of residues that could be mutated, and the positive cooperativity of the modulator with the endogenous agonist adenosine was lost, suggesting that these are contributing to the binding of this ligand. We also validated this through a bit of docking and a bit of MD simulation. So overall, quite a lot of work went into sort of validating the location of this binding site, as at the time it was relatively unusual. Now, sort of the bread and butter of our lab has really been muscarinic acetylcholine receptors. Since I joined the lab in 2011, it's probably been one of the main receptors that we've been working on. And that's because they're an important therapeutic target. And the M4 receptor has a strong role in schizophrenia, and activators of the M4 receptor are being pursued as potential treatments for schizophrenia. And I think this is probably more relevantly shown here by recent clinical validation of activators of the M4 receptor. So this orthosteric ligand xanomeline has recently been used. So as previously, historically, xanomeline is a very interesting compound for a number of reasons. One was it had clinical efficacy in phase 2 trials, but failed because it had a lot of off-target activity at other muscarinic receptor subtypes. So the solution to that was if you co-administer it with a peripherally restricted muscarinic antagonist, you could sort of block the side effects it was having by activating muscarinic receptors in the periphery. And this treatment is called KarXT. It does have clinical efficacy, and it just met its benchmark in a phase 3 clinical trial. And so I believe they're trying to seek approval from the FDA to use this as a treatment for schizophrenia. Similarly, other people have been developing M4 positive allosteric modulators, and this is an example of an emraclidine, which just passed the phase 1b trial and now is going into phase 2, and it binds theoretically to an allosteric site that is distinct from where xanomeline binds. And so we've had a long-standing interest in trying to understand how these ligands work, both pharmacologically, behaviorally, and of course structurally. And we've been quite fortunate in that we were able to determine a number of structures of the M4 receptor bound to different orthosteric agonists, such as the endogenous ligand acetylcholine, a superagonist cyperoxo, as well as a couple of positive allosteric modulators, VU154, LY298. These are M4 PAMs that are selective and have sort of been instrumental in sort of the preclinical validation of this target. I'm not really going to talk about these today, as that's a story for another day. Another interesting story that I won't talk about, but just briefly mention, is work on a new M4 PAM scaffold, XY6, which we also were fortunate to get a structure of. It binds in a slightly different shape and conformation in comparison to the other M4 PAMs, and the medicinal chemists have done quite a lot of interesting SAR around this compound, and then brought in some new insights there. Instead, I want to focus on a structure that we got with the xanomeline, which is the orthosteric agonist that is now progressing potentially to treat schizophrenia in humans. And one of the surprising things when we got this structure using Cryo-EM was that xanomeline was indeed bound in an orthosteric site, but it was also bound in the extracellular allosteric site. And that was something that was sort of a bit of a surprise. Shown down here, you can see clear EM density for the orthosteric ligand. And then sort of shown up here, you can see clear EM density for the allosteric ligand. Now the orthosteric ligand, there's actually a lot of cool things happening here. And that typically the agonists of the M4, such as acetylcholine or iperoxo, are relatively small, and they only partially fill this orthosteric binding site, whereas xanomeline has this hydrophobic tail that kind of inserts itself upwards into this sort of sub pocket, and makes a really nice fit into the orthosteric site. But it's also bound kind of in the allosteric site, and this is in a location that overlaps with where the other M4 modulators bind. And we thought this was a bit unusual. Having said that, xanomeline has a history of being an unusual compound. So we wanted to try and validate this further. just to convince ourselves that xanomeline could potentially be binding allosterically. We were fortunate to do some liquidynamic simulations with Runjor at Stanford. They kind of had, there's a whole other project involving xanomeline that we've been working on them with, trying to understand why xanomeline has functional selectivity at the M4 and not the M2, which is a story for another day. But what they did was some MD simulations where xanomeline was bound in the orthosteric site, and they looked at the binding of xanomeline to either the allosteric site, the PAM LY203298 to the allosteric site, or the agonist hyperoxo to the allosteric site as a control. And when they did hyperoxo, what they found was that it could indeed bind to the allosteric site, but that was only for a brief duration of time, and then it would unbind and move on. If you take the PAM LY203298, it actually bind to the allosteric site, and when it bound, it generally stayed bound for a long duration of time during the simulations. And interestingly, xanomeline sort of behaved the same way as LY. As once it bound, it would stably interact with the receptor, suggesting that xanomeline could be binding allosterically. We turn to some pharmacology assays. This is an assay looking at the dissociation of the radioligand NMS with increasing concentrations of xanomeline. And if the ligand is allosteric, you might expect it to slow down the dissociation rate of the orthosteric agonist. And as that is indeed what happened with xanomeline. As we dialed in increasing concentrations of xanomeline, we'd see that the rate of dissociation would slow down, suggesting a potential allosteric mechanism. And we could also mutate a key residue that sits in ECL2 nearby where xanomeline binds, F186. We can mutate this to an alanine, and we would abolish the allosteric effect of both our prototypical M4 PAM, but also xanomeline. Now, one could argue that this was predicted by Arthur Christopoulos back in his postdoc days in this paper in 1997, where he sort of drew xanomeline binding to the orthosteric site of the receptor, but also this other site, and this was called an exosite. And they didn't really think it was allosteric in the sense that I've been talking about allosteric ligands, but rather xanomeline has some strange pharmacology. In particular, it has it's wash resistant, so you can treat cells with xanomeline, wash. Xanomeline will still be bound and persistently activate the receptor. So they thought that potentially xanomeline could be binding into some sort of exosite on the receptor that sort of stays there and can activate the receptor. We've been trying to see if this sort of exosite is linked to the allosteric site, and our suspicion is probably not. It's probably due to the lipid tail of xanomeline sort of inserting itself into the membrane, and then maybe on the way into the orthosteric site, it kind of stops stably in the allosteric site before moving on to the orthosteric site. But overall, a very strange ligand, and it's quite fitting that this would be one of the first examples where a structure of a GPCR has been determined with a orthosteric agonist bound in the orthosteric site, but also in an allosteric site. And this is a story that if you're interested in, I'll present the more full version of this at ComBio at the end of this month. We haven't just been working on the M4 receptor, we've also been working on the M5 receptor. It's sort of black sheep of the muscarinic receptor family. It's last to be cloned and certified as a member of the family. It's also the least to be studied. It has been implicated in a number of physiological, has a physiological role in a number of CNS disorders such as Alzheimer's disease, schizophrenia, and drug addiction, but there's been a need for more selective M5 ligands. And so this is work that kind of goes back to Patrick Gentry when he was at Vanderbilt as a PhD student. He helped discover probably the first M5 selective PAM, ML380. And so M5 PAMs are potentially interesting for the treatment of Alzheimer's disease and schizophrenia, whereas M5 NAMs he also helped discover and are potentially useful for drug addiction. And so he came to MIPS and wanted to try and find what makes these modulators selective. Can we try and identify where they bind? This project sort of started at a time when we were relying heavily on X-ray crystallography. we were able to generate a large number of structures with different allosteric modulators bound, I should say, in the presence of a number of different allosteric modulators. Unfortunately, we were never able to see electron density anywhere indicating that these ligands are bound, and that included adding sort of heavy atoms so that we can try and do anomalous diffraction experiments to isolate if these might be hiding somewhere. So we instead turned to mutagenesis, and Patrick mutated a lot of the key residues in sort of the traditional M5 allosteric site, and found that really mutation of any of these mutation of any of the residues in sort of this common allosteric site didn't really affect the pharmacology of ML375 or ML380. So we were a bit stuck. Fortunately, with a bit of time, we started doing a bit more molecular docking and trying to dock ML375 into sort of the various sub-pockets around the receptor. And we sort of combined that with a refined mutagenesis approach, and we were able to find a small pocket just outside of TM3 and 4 on the outside of the receptor, that if we mutate 3 conserved residues to alanine, we could completely abolish the activity of ML375. So we thought that this is potentially the binding site for ML375, though that's something that we'd like to eventually confirm through structural approaches. At the same time, we sort of wondered, well, what about ML380, the M5 selective PAM? Is it sort of speculated that it might be binding nearby ML375? And if we actually, so this is showing at the wild type M5, ML380X is a PAM, changing the dose response curves to the left. If we use this pocket that had the 3 residues mutated to alanine, ML380 had full activity similar to wild type. So that suggested that this was not the location of ML380. So we sort of turned to Cryo-EM to see if we get a structure of this. And in the last 2 months of Wes's PhD, while he was writing up, he was able to get a Cryo-EM structure of the M5 bound with iperoxo and in the presence of a high concentration of ML380. And I've sort of left a question mark here as we've maybe identified the ML380 binding site. When we were looking at the maps, we saw sort of 2 regions of EM density that clearly stood out. One was around the top of TM1, 2, and TM7, and this was a region that I suspected is a lipid, as it's kind of a bit of density that we see even in some of the X-ray crystal structures of the M5. It was just more clear in the Cryo-EM, so I suspect there's a lipid that might be hanging out here. But nearby the region where we thought ML375 was binding, we saw this nice, well, I'm not gonna say nice, this set of density that looked like it could be ML380. And we could kind of fit the ML380 into this density and see that it could make a number of key interactions with hydrophobic residues that were nearby. So we're sort of calling this the putative ML380 binding site for the time being. This is obviously to be confirmed by further, hopefully, structural evidence and pharmacological evidence. And this is sort of at the bottom of team 2, 3, and 4. And this sits just below, we think this sits just below where ML375 binds. So this is where we think ML375 is binding based on docking studies. And this density is very, very clearly sitting just underneath that region. It's also possible that this could just be a lipid, as lipids do tend to bind everywhere. What we do know, though, is if we make a chimera of the M5 with the M2 receptor, and ML380 doesn't really have any activity at the M2, and we make a chimera with TMs 2, 3, and 4, is we can completely knock out the activity of ML380 as well, suggesting that it is probably somewhere in this vicinity. I mentioned the previous bit of density around TM1, 7, and 8. And it's likely that is not ML380, as there's no difference when we make a chimera there as well. So we do think we've identified this binding site. It does somewhat overlap with lipids from other GPCR structures, and that could be why we're having a hard time getting good resolution on the density of this ligand. And truth be told, we really just need better, more soluble versions of this compound to probably try and facilitate future structures. And then sort of the last example that I was going to talk about was really one last example, and I was actually really excited about this example, is that it was really an example that was kind of at the bottom of sort of the ups and downs of structure-based drug discovery. And it was an example where we're able to get relatively high resolution GPCR structures, both in the absence and presence of a positive allosteric modulator. But unfortunately, we were not able to ever identify the location, no hint of density anywhere. All the maps looked identical. And so we were trying to come up with a story on publishing these structures as they're still relatively high resolution, and some cool stories could be made from these. We sort of had one last go with the help of some chemists. A more soluble version of the positive allosteric modulator was made, and that actually facilitated the structure that we could identify the PAM binding site. Unfortunately, that new, more soluble PAM has some potential IP associated with it, and so decided that we'd probably just not show that ligand in this presentation and talk about this example less. But nevertheless, I wanted to include this, as this was an example that when I submitted the abstract 4 weeks ago for this talk, we really were sitting at the bottom. We didn't have this new structure. We really just had these 3 structures, and it was really the medicinal chemists that were able to pull this project from sort of the bottom all the way up to the top, and hopefully we can share where this story goes in the future. In terms of future directions, we're interested in trying to combine the Cryo-EM structures with some of the various SAR efforts that some of the medicinal chemists that are using, and combining these with more differing computational approaches and screening approaches to try and identify new scaffolds and ultimately design better therapeutic molecules. I'd say there's a number of current challenges that still exist in the field. And I think the Rolling Stones probably said it best, and you can't always get what you want. But if you try sometimes, well, you might find, and that's, you never really know what you're going to get until you try. If you try and still fail, be nice to the chemists. They're your friends, and almost all of our work wouldn't be possible without their support. And so we're very thankful that we get to work with a lot of really good chemists, a lot of really good people in general. And I think one of the main challenges at validation of small molecule binding sites, particularly for membrane proteins near detergent micelle, can be a challenge in Cryo-EM maps. And I suggest using a caution when doing so. And it's important to include as much experimental validation as possible. And hopefully better tools will come along in the future that help with the validation of these sites. There's already multiple PDB entries that likely contain ligands that have been incorrectly modeled into noise, the bid and micelle, and I suspect these numbers will increase as more and more people are excited to try and show where their favorite molecule binds into the micelle of a receptor that likely has a lot of its own lipids occupying as well. And that's not to say that X-ray maps aren't problem-free either. They have these similar challenges as well. And with that, I just want to say thank you. This work that I presented has been the work of a lot of different people throughout our lab. In particular, Jiva, Jesse, Wes. They've done a lot of work, particularly with the help of Matt. Patrick and Arthur and Celine have all been instrumental in getting these projects forward. In particular, Elisa as well. Got a bunch of fantastic collaborators, and of course, this wouldn't be possible without support from the NHMRC, ARC, Wellcome Trust, and the CCEMMP Center. And with that, I'll take any questions. Thank you.
Thank you, David, for an amazing presentation. It's like very interesting to know about all the allosteric modulators, the ligands. So the panel is open for questions. Anyone who has any questions, you can just unmute yourself. And so we see Patrick. Yeah.
Thanks very much, Dave. Can you hear me?
Yes.
So I mean, really nice talk. Some really super interesting work going on. Going back to your P2X1 channel, was that in an open or a closed state, or can you tell?
Yeah, so we think it's in a desensitized state, and that the channel is the pore of the channel is too small for ions to pass through in comparison to P.
Yeah. Did you think that you can manipulate that to get the open states as well?
Yes, but it's tricky. People have done it before, and it involves making chimeric swaps between receptors in the TM region. It's something Felix is already actually cloned and is going to have a go at. It will require a validation using a little bit of electrophysiology, which is something we haven't really done, but we'd probably seek out to do. Is it'd certainly have an impact.
Oh, very nice.
But in the meantime, we think we've gotten a ligand-free sample that hopefully this week will validate on the microscope. And that's just doing extensive dialysis.
Mm-hmm. And have you had any luck with complexes with any of the novel ligands?
That was supposed to be actually screened today, but might be screened tomorrow. So stay tuned if you.
I'll keep my fingers crossed for you. Thanks, David.
Thank you, Patrick.
Any more questions? Gogan has a meeting, so yes.
So, oops, I have another one if nobody else is asking.
Yeah.
In terms of, where, you've done lots of things to try and, identify ligands, for example, with lipoxygenase project, is there anything that can be done sort of at a chemistry level to potentially make a covalent version of these that might give you some guide to where they're binding?
So you'd asked me about covalent inhibitors for some of the GPCR projects, in which I'd say the answer was yes. We've been thinking about that for 12 locks. I think that's probably the next step. They have different chemical, they have different small molecule inhibitors. The problem with this one is I suspect it's not the most, despite the fact that it's going into humans, it's not the most soluble inhibitor. And they have to add a lot of PEG additives, I think, to give it to humans. And so I think solubility is certainly an issue, and we do think there are other small molecules that could potentially could be useful, but we haven't really investigated the irreversibility aspect of it. In part, doing that with lipids is well, with lipid molecules, it's a bit more expensive. With the small molecules, that could probably be done. It's just not something we're trying to more explore other avenues where we could purify this enzyme in a manner that doesn't have the CoA substrates bound, as we think that's probably the major challenge. And that could be something that's just unique to purifying from mammalian systems, whereas if we move back into maybe insect cells, maybe that'll be less of a challenge. But it's something that we're definitely thinking about doing.
Cool.
We haven't done it yet, but
from other people.
Oh, good.
There you go.
Oh, we have a question from the Acro. What was that?
That's a lot of people.
Yeah. Could you please unmute yourself and ask the question? All right, yeah.
All right, are we unmuted? Hello?
Yes.
All right,
Hello.
Dave, your M5 structure with the positive allosteric modulator, I've forgotten the name of it, the one that looks like it binds at the cholesterol binding site. Yeah. Have you tried sort of phase improvement methods to maybe improve the density around that area? So there's a few different methods like log scale or just standard crystallography phase improvements. Have you tried any of these sort of methods to maybe identify it?
No, not yet. We've done a bit of hoping that sort of similar to 12 blocks, we could do a bit of 3D variability analysis and try and pull it out from there. But so far, it's been a rather ambiguous binding site that we've been modeling into and really haven't had any luck at improving.
Yeah, because there are ways that you can calculate sort of emit maps. I know it doesn't make that much sense because you've already collected phases directly, but it can be helpful.
Yeah, no, we've just started thinking about trying to use a few of those programs, and it would be interesting to see sort of what the resolution requirements are around having interpretable data from those sort of methods. But it's a good suggestion, one that we're certainly going to try.
Cool.
I think the simple question was really brought up by Patrick is trying to make it reversible. There's a really nice cysteine that sits just next to where the ligand binds. So I suspect that if it's a chemist, we could probably make a version that will irreversibly react. Or just make a version that's more soluble. That'd be better.
Hi, Matt. Hi, Matt. Can you hear me?
Hello.
Can you hear me?
Yeah.
Yeah, it kind of got the proper answer, what you were looking for?
I don't know. yeah. David had
Anyone else want to ask something or discuss something?
No, we're good.
Yeah, Thank you, David, for the amazing talk. It's really interesting.
Hopefully
thank you
there'll be
all
more
for joining us today, and the presentation is already recorded. You can find it on the center website.
Well, maybe soon, because it needs to be uploaded. But yes.
Sure, Patrick.
It'll be this afternoon, probably. There's some more bits to happen before I can do that.
All right. Tracy would know that better.
It will be up.
Yes.
Thanks very much, Dave.
Cool.
Thanks,
Thank
Dave.
you, everyone.
Thank you.
Have a good one.